Okay, grade 12, as requested, this is your experimental design kind of review. This is part of a unit I usually do in grade 11 to kind of help students refine their design skills and get ready for the IAs. So I've kind of taken this and eliminated things that aren't relevant for your exam, and I'm focusing more on general skill sets. So hopefully this will help you prepare for your exam, because you will definitely have to talk about how to design an experiment, what test to choose, that kind of stuff, because that was a big part of our course this year with your IA. Okay, so first thing, statistics is really important in science. Is how we convey meaning to our data. And it's not just taking a mean, taking an average, which hopefully by this point you guys know that very well. So for the IB, what they actually need you guys to know is this list here. Okay, mean, median, mode, range, different kinds of graphs, probability and p-value. The t-test will not be on your exam, but you'll be asked to kind of choose which test would work. Chi-square will be on your exam. Standard deviation, you need to understand what it means, not calculate it necessarily, or not calculate it at all. And then causation and correlation. That's what it says in the IB subject guide for this. That's what your exam will be based on. Okay, so why does stats matter? Stats is the grammar of science. It's how we look at science. It's how we understand our data and if the variables are related or not and what it actually means. So I figured out how reliable our data is and how we can tell how reliable the data of other scientists is. So in the information age we're in right now, there's so much information available online to us. With the whole COVID thing, it's a really great example. You need to be able to look at studies and look at the results given to you and figure out, okay, how reliable is that? How many trials were done? How was there a control group? Were there good constants? Is it repeatable data? Is this valid? Before you make decisions. And we hope that all the leaders in the world and healthcare professionals have a good understanding of statistics. That way they can understand what people are doing, because you can really manipulate the numbers in stats to kind of show what you want to show if you're not careful. Or if you are careful. Okay, so the first kind of stats, the simplest one, is descriptive statistics, which are the tools we use to describe a data set. And the sample is your data set. So we'll be using stats as sample. And you can figure out if you have sufficient data for further analysis or not based on the descriptive statistics. So let's look at some different examples here. Mean, median, mode, and range. Very simple things you probably learned about in lower grades. But they actually are very useful for telling if there's a really weird thing happening in your data or not. Okay, so mean, of course, is the average. The median is our middle value. We write the numbers in order. So here in little people, we have the height written in order. So my median is the center value. If I have two values, I have an even number, then the center value will be those two, then divided by 2. The mode is the most common number, so the mode here is 155, the two people who are 155 centimeters tall. And the range is the spread, so the range for this one is from 155 up to 168. So we need all 4 of these because they all help convey different information and gives a clear picture of information. So if, for example, the mean is very different from the median, that's a big sign that something is different here. There's maybe more, the data is more to one extreme or the other and not really reliable. Okay, a good way to do this is look at hand length. This example, if we did this, actually I would do this with my students this year and did analysis. Okay, so when you're actually planning your experiment, and this will definitely be a part of what you have to do for your exam, so be prepared. You have to plan with the investigation in mind, with the data in mind. What are you trying to prove? What do you want to mean? What are you hoping it means? So the quality and quantity of the data is a big difference between like a middle school, grade 9, grade 10 lab report, where someone is just kind of learning the basics of how science works versus what's going to be expected in the DP. So you're going to keep track and keep in mind the validity and reliability of your data when you are designing your experiment. Designing your methodology, which of course methodology is kind of the philosophy, the principle behind your experimental design. So validity is, does your test measure what you want it to? So if you look here at the target analogy, this is both reliable and valid. Okay, if validity is the target is what you want to measure, that is center here, right? So this is not valid. This is kind of valid. It's close to center. This is not even close to the center, so it's definitely invalid, and this is on the center, so it's valid. It's measuring what you want it to measure. And then reliability is how consistent is it? So if you're consistently hitting that target, that's going to be reliable. This is reliable, and this is also reliable. So it's really important that your data is both reliable and valid. It's measuring what you want it to measure, and it's consistently measuring that in a similar value. So there's not a ton of sources of error messing up your data. So how do we make sure something is valid? Well, one thing you can do is constants. You want to make sure that everything that could affect your independent variable, your dependent variable is kept constant except the independent variable. That's the only thing that should be affecting it is the independent variable. Everything else should be kept constant that could possibly affect it. Now, sometimes students go too crazy with constants and they list things that have no impact on them whatsoever. That is not helpful. You need to make sure you're only focusing on things that actually are going to have an impact on your results. Another good thing to do is to use a control group and an experimental group. In the control group, you don't include the independent variable, so you can see if something else is affecting your results. So if your validity is what you're designing is affecting what you hope it's affecting, so you can see the results on the independent variable, then a control group lets you know if that's the case, because you're not including your independent variable. So if the dependent variable still changes and something else is there, you forgot a really important constant. So here's an example. We have two groups here. The independent variable is a medication, and the dependent variable is a response in the participant. It's medical testing. In the control group, people are going to get the placebo, an inactive medication, to see if there's some sort of effect, maybe something else is causing this change going to happen. And then the independent variable is the active medication, so this is the experimental group. But both of them are measuring the dependent variable to see if it's being affected. If this group with the placebo is changing in some way, then something else is happening here. And that's a way to make sure that this part of the experiment is not affecting things the way you think it's supposed to affect things. Okay, so here's an example experiment. So Joe wants to determine how increasing amounts of growth hormones affect the mass of pea plants. So his independent variable is the amount of growth hormone, dependent variable is the mass of the plant. So how can he make sure his experiment is valid? Well, he can do constants, and some good constants for this would be things like the amount of water, and frequency of watering, starting the plant size or age. Are all the plants going to be from seeds? Are they all going to be from the same initial kind of mass? How's it going to start? The amount of fertilizer being used, even if it's none. The sunlight exposure and intensity, no predation or parasitism, the length of time allowed to grow, all those things are important constants of this experiment. When it comes to things like plants, what you can do is you can either keep a very controlled environment, like in a lab, or if you can't do that, you can put them all in the same environment outdoors, which will be variable, but it's variable in the same way for all the specimens. Either one of those will help make sure your experimental results are valid. You need to have a control group, which has no hormones given to it. An experimental group has different amounts of hormones. Several different groups. Okay, what about reliability? So the best way to make sure it's reliable, that you're getting the same repeatable results, is that you have many trials. 3 is kind of the bare minimum what we have students do in like middle school, grade 9, grade 10. 5 is okay, even more is better. Another part of this is if you have many different individuals. So for most animals and more complicated organisms, 30 is the minimum you need. But like in medical studies, there's many, many hundreds of thousands of specimens of individuals included. And that's because there's just too many variables to control. So you need to have lots of sources of data to help negate that effect. Oh, sorry guys. Okay, a final way to do this to ensure reliability is when you publish inside a community, you need to make sure your procedure is repeatable. So some other scientists can do your experiment and see if they get the same results. A big part of how we ensure reliability in science. And before a new idea becomes a theory, becomes part of kind of the scientific knowledge base, it has to be tested by multiple scientists and reported on. Okay, so back to Joe's experiment here about how growth hormones affect the mass of pea plants. How can he make sure it's reliable? Well, he can include at least 30 plants in each group, 30 plants in the control group and 30 plants in the experimental group. 30 is a little bit is higher for plants, but the more the better. I would say at least 10 for a plant study in each group. He can then repeat the entire experiment 3 or more times. So the total, if he did 30 individuals in each group and he did 3 trials, would be 90 control plants and 90 experimental plants. That is a lot of plants. And that's how you make sure your result is reliable. This is why a lot of these big studies take years. He can also write his procedure very detailed and specific so that his experiment is easily repeatable by other scientists once they publish it. Okay, another part of this whole process is doing a pilot experiment. Because biological experiments are really much harder to control than chemistry or physics experiments, just because you're dealing with living things, and living things have so many other variables, a lot of times we do pilot experiments in biology. So we can't keep everything constant in an organism really well, so what we do is we do a pilot, which is a short-term with less sample size experiment done just to double check your methodology. So you may have thought really hard at a bunch of research, made the best possible methodology you possibly can to test your particular experimental design. But if you don't do a pilot, you may realize you forgot something you just didn't think about. And if you do a pilot, you can take a few days, get some data, you might notice some errors, and you can change, make small tweaks to your methodology, and all of a sudden be much better in the end. So it's a good thing to do that in biology. Okay, the other thing is when you're designing an experiment, you need to keep the kind of statistical tests you're going to do in mind. So you can't go back, if you wait to the end of the experiment, you have all your data collected, like, okay, so now I'm gonna choose my stats test. You are unlikely to have sufficient data, the right kind of data to do the ideal kind of test for your experiment. So a lot of them have minimum requirements, 30, at least 30 for a lot of them, sample sets. So if you make a choice of which kind of test you're going to do before you actually collect the data, you're going to make sure you have sufficient data at the end. Okay, if you can't find a test that works, then you need to change your experiment. Your experiment is not well designed if none of these tests will work. So the kind of tests that are often used in biology, if you have frequency, so you count only chi-square tests, we're going to talk about that one. This is compared between observed and expected. We did that in ecology. Then you have the unpaired t-test, the ANOVA, and regression. So this one you have to be able to do for your exam. These 3 you don't have to, but it's good to know that they exist and are tools you can use. So I might ask you to design an experiment, what kind of statistical test you would use. Okay, so we have different individuals here comparing 2 sample means for a t-test. In ANOVA, you have more than 2 groups, and you're analyzing the variance. In regression, it's from your graph, right? A scatter plot. If the relation is linear or nonlinear, you're looking for association between the 2 variables. Okay, so it's good, and put this in your notes. Open notebook test, guys. Put this in your notes so you can make sure you're picking the right test. Okay, let's talk about graphs. So we might have collected hand data, and then you have to graph that. So here are the 4 kinds of graphs most often used by scientists, and the ones I want you guys to know. So bar graphs, line graphs, scatter plots with a line of best fit, and histograms. But you have to pick the right graph. A lot of students struggle picking the right graph. So here's our graph types. Okay, why is this the graph type chosen so important? Well, it shows you the pattern in the data, and if you don't pick the graph that best communicates that trend in the data, you're not going to notice things like outliers. Like if this data here, if I had this as a bar chart instead of a scatter plot, you wouldn't see all these outliers up here. This is much more informative than this would be for that. Okay, so make sure you're thinking about which bar chart works the best. So line graphs, review of what line graphs are. We have our x and y value. For every x, only one y, and those points are joined by straight lines. Usually your x-axis is your independent variable, y-axis dependent variable, and you might use a line graph in a situation where both variables are quantifiable and they are related and not discrete. Like a change over time is a really common one to use a line graph for because time is continuous, so you can join those points with a continuous line. Okay, bar graphs. We use rectangles to show the amount of data within a certain category. So this is the case where you have a number on one part and a category on the other. Okay, so the y-axis is quantitative And the x-axis is qualitative, so it's using words, not a number value. And we separate them by empty spaces because they're separate things. Okay, in histogram we connect them because it's frequencies. Here we leave a space because they're separate things. There's no real reason why it has to go in a certain order for the bars. It can be any order, but it just has to. It's not going to change the trend, but you have to make sure you communicate clearly what those are, right? Here's a key, and there's a key for that one. So the kind of data we would show on a bar graph, so you have maybe different parameters. Here are some different concentrations, different chemicals, and how it's going to affect citric acid and consumed sugar produced. This is an IB exam question. Okay. Scatterplots, where you have two variables that are involved, that you apply one x and y value. So for every x value, you have one y value. But you have more than one x value, you can have multiple y values. Oops, that's a typo. Okay, so there's multiple ones can show up. So it's good to have a scatterplot for that. So you can see here I have several that have the same x value. Here are very similar x values. They're kind of stacked on each other, and it shows you all the variance within that data set. Sometimes you put a line of best fit, but you never ever connect the dots because they're not directly related. It's not happening over time. Connecting the dots is a different type of graph completely. This is showing that they are a pattern, but it's not related to the same individual over time. Okay, so here's when you have both variables are quantifiable, but they're not related. Like we're comparing different individuals, a scatter plot is the best one. Okay, histogram, they use rectangles to represent the data like a bar chart, but here we have some sort of quantitative scale on the x and we have frequency on the y. Okay, so time or age would be a good one for this. And then the width of the rectangles represents the incremental values. There's no spaces between them unless you have a value of 0, like here there's no one between 80 and 90. So show those continuous values. The intervals are something called bins, and so you can think of it as like how often that particular bin was used. So the frequency is between 20 and 30 was used 2 times, between 30 and 40 was used 4 times, between 50 and 60 used 5 times. Okay, so what kind of data use in a histogram? I think we were comparing population kind of frequency. So between these ages of 30 to 39, here's the dietary fiber quantity. The total, and looks like here might be a mean with their deviation. So this one is showing a histogram and kind of bar chart combined, which is kind of cool. Okay, let's be careful about correlation versus causation. I really like this cartoon, it's pretty funny, so take a look. So correlation is defined as a relationship between two variables such that when one changes, the other does also. So in this cartoon, which two variables are correlated? Well, sales and shaving heads. And this correlation can be positive or negative. What does that mean? Positive or negative correlation? Well, positive if one increases, the other increases, and negative is as one increases, it decreases. So the statement, correlation does not equal causation. What does that mean? Look at the little cartoon here as an example. So even though there's a correlation between sales and shaved heads, that doesn't mean that you are going to be more successful at sales if you shave your head, right? It's not, shaved heads don't cause better sales. Maybe people who have shaved heads tend to be more confident because they're confident enough to shave their head, and that confidence is a big factor in being a good salesperson, so it increases that way. There's some other factor probably affecting it. Okay, so types of correlations, you have positive, strong positive, weak positive, strong negative, weak negative. Look how things are not really on the line as much in the weaker ones. A moderate negative, and there's no correlation at all here. Okay, so interpreting graphs happens a lot in the IB. You'll definitely have some of these in your exam. You just had some on your assignment. So the key things here is what information is included. So what does the key say? What does the description say? What do the axes say? What information is here? You have a title, axis labels, a legend or key, and you can look at the trend and look for what's happening. And the IB likes to give more complicated ones, so it's not. This is not an easy to see trend, right? you'll notice the woods numbers, the little X's, are mostly further down in percent of yellow base color shells, and then in the fields, it's more up here, more of the yellow in that one. Yellow and unbanded is more common in the fields, and yellow and banded, sorry, and yellow and unbanded and less yellow base is more common in the woods. Okay, another graph you can interpret. So IB loves these kinds of graphs. This is the full-size one. We have the actual intake as a mean and the recommended intake for different males of daily dietary fiber of males and females. It's a combination of histogram and bar chart. Okay, another one, here we have a buildup of sodium chloride in the soil. So the question IB would be outline the trend amount of salt. So here we have a negative correlation. As time increases, the amount of salt decreases. Okay, another question here about cancer in this case. Align the relationship between the fiber intake and colon cancer and mortality. And actually interpreting graphs to determine the predicted mortality due to colon cancer. Okay, further interpretation of graphs. More graphs. Here we have day length. Just pause these and practice reading them. This is a really more complicated one. So again, take a practice, reading them, interpreting what it means. Okay, so we have linear and nonlinear relationships as well as positive, negative, strong, weak correlations. So in a linear relationship, the data tends to form a line. It can also be a curve, like an exponential curve. Nonlinear, it's not following any known pattern. Okay, so here we have linear, nonlinear. So linear and direct and inversely proportional. So if it's directly proportional, that means as one is increasing, the other is increasing. If it's inversely, as one is increasing, the other is decreasing. Other good words to use to describe data. Okay, when you're looking at an entire data set, you often will look at the normal distribution. And it's one of the most common ways to look at it, and where you see the most common value, how close are to the mean. Okay, so here is the mean in that particular data set, and if you go below the mean, it gets less and less common, the frequency less and less until you get to 0, and above the mean, frequency decreases until you get down to 0 again. So it's more and more common to be at the mean, whatever that data set is going to happen to be. Okay, a lot of variables follow this shape of distribution. People's height, blood pressure, a lot of physiological variables follow the distribution, the number of leaves on a certain species of plant, the number of hours of sleep someone gets, these would all mostly follow this distribution. Normally we have a symmetrical distribution in which we're going to have the mode, median, and mean are all the same, but an asymmetrical is not going to be the same. And that's a good sign that, okay, if I do my descriptive statistics and these are not the same, then something's off here. Okay, here's another old IB exam question where you had to calculate how much lower the mean mass of nematodes is in these different samples. So again, give it a shot. It's good practice. Okay, and here's the answer key for that one. So check your work. Okay, standard deviation. You need to be able to interpret what this means. So standard deviation is a measure of how spread out the values are. So we normally say that one standard deviation is where you're going to have 68% of the values are within those first parts of the bell curve of the normal distribution. And then 95% are within 2 standard deviations, so 2 above and 2 below. 9.7 are between 3 above and 3 below. Okay, so if you have a data set, the more of your data that is in the standard deviation, the more similar data is, so the more reliable your data is. If you have a really big standard deviation, that means that most of your data is very spread out. It's not following this kind of curve, and there's probably something unreliable, maybe invalid about your design. Okay, so you don't need to figure out how to calculate this. It won't be on your test to calculate this. You need to be able to interpret it. Here's an example question. What I have here is the two populations. Calculate the mean value, and then explain what are the terms standard deviation. Okay, error bars. A lot of time you see error bars on IB exam questions, and a really useful tool to use in statistics. And what they do is if you're plotting the mean value, we often include error bars, and error bars show usually one standard deviation above and below the mean. So the bigger the error bar is, the more variance there is in that data. So this is a very small error bar. That means the standard deviation is very small between above and below the mean. So most data is very similar. Whereas these ones have very large error bars. There might be some sources of error there, maybe some outliers, something causing a difference in your data. Okay, so if you look here, when the error bars overlap on a graph, it means that the difference is not statistically significant. So if there's an overlap, that means there's means between the two of them. So this means the difference here is probably not significant. And here it's probably somewhat significant, but maybe closer to than this one. And here it would be significant. Okay, but to be sure, we have to actually do a statistical test. We can't just say that. Okay, so here's more. Look at these big error bars. You can see the very small error bar here compared to how big these ones are. These two, there's no significant difference, and these two, there is a difference. There's no gap in no overlap between error bars. You can also include error bars in line graphs. Another graph with error bars. So practice interpreting all these. Okay, so let's talk about significance. So what does it mean for something to be statistically significant? Well, what it means is the relationship between the two variables is caused by one variable is causing the other. It's kind of giving that we're 95% sure of causation, despite any possible sources of error. That is due to the error and not for chance alone. Okay, so we have to figure out doing that. One way to do that is a t-test. You can also do a Pearson's correlation. The p-value we use for this in biology is 0.05. So I find this to be a really funny cartoon because I know people who've had that issue where their data is so close to being significant but not quite there. So if it's not significant, we cannot say there's a relationship. And a lot of times experiments that maybe the averages look different, the means look different, and the graph looks different, and we interpret it in lower grade level as being a pattern or supporting your hypothesis is not the case, and you actually do a statistical test. Okay, so probability of the p-value. Probability means that the chance something is happening. It's how likely it is to happen. So the p-value is a probability that chance alone could produce that difference. So the p-value in science is 0.05%. 0.05, which means that you were 95% certain that that difference was not caused by chance. That difference was caused by the two variables. So if you have your observed data here, you're allowed to have some off ones here and here. They're going to be your p-value. Those 5% on 2.5% on each end, that could be caused by something else, but you're 95% sure that this was caused by your independent variables affecting your dependent variable. Okay, so the likelihood of observed data occurring if the experimental hypothesis is correct, that's your p-value. And if it's less than 0.05, that's the likelihood if the null hypothesis is correct. Okay, it's due to a relation between variables and not chance. If your p-value is less than 0.05. The smaller your p-value, the better. That means you're more certain that your data is caused by the two variables relationship. Okay, so let's look at the null hypothesis. So null means there is nothing. Nothing is there is no relationship. In the null hypothesis, we symbolize it with h0, h0. It states that there is no statistically significant difference between the samples. So let's say the sample here, I have two goldfish samples, and it's about crowding or population density of goldfish and how big the goldfish are. So in this experiment, the null hypothesis would be there is no difference in mean length between the denser and less dense populations. So my null, if there's no relation to my experimental, could be something like there is a difference in mean length between the denser and less dense populations. Okay, so percent change is a good simple test to do, especially if you're doing things like the Joe's plant experiment. Instead of having the exact same mass of plants to start with, which would be really hard to do, you can instead be comparing percent changes over time. So the nice thing here is it's better than a difference in value. So our data points, if they're not exactly the same to start, it makes them more comparable by showing how much they've changed versus just what their initial value was and final value was. Okay, so if you think of the potato core or dialysis tubing labs, this is really helpful for those kind of labs because not every single potato core weighed exactly the same. Even though you used a cork bore and made them exactly the same size, in dialysis tubing lab, they weren't all the exact same volume or mass to start off with, so doing percent change is a lot more indicative of the actual change and what the actual relationship is between the variables. So since you can't really control all parts of this, it's better to use percent change rather than the difference between. Okay, let's talk about chi-square. So chi-square allows us to calculate how closely our observed results are to expected results. This will be on your exam, so be prepared to do a chi-square test. So we can see if it's caused by chance or some other unexplained phenomenon. A great example of doing this is for Punnett squares. So in a Punnett square cross, you can expect them to be Mendel's ratios, so 3:1 for two heterozygous parents, or 9:3:1 for a dihybrid cross of two heterozygous parents, and you figure out your expected from the actual outcomes. So here's I like to do chi-squared in a table format. I find it very helpful for me, but you can do it other ways as well with just using the straight formula if you prefer. So here's a formula where chi is the Greek letter chi for chi-squared. O is the observed values, which are the actual results of an actual experiment. E are the expected values, which you calculate theoretically. And then sigma is the sum of. So when I'm going to do this, so just like almost any statistical test, you have to use a critical values table first to figure out what the chi-squared value is. Okay, and then you can figure out how many degrees of freedom there's going to be. So for chi-squared, you take the number of categories and you subtract 1 to figure out degrees of freedom. And you also need to know the accepted p-value in your field. So for biology, we know the p-value is 0.05. Okay, so here's an example. So we have a monohybrid cross of smooth and wrinkled seeds. We have a data of the P generation, F1, and F2 generation. We have actual results here, the observed results. So here's my observed. I had this many smooth seeds, this many wrinkled seeds, and there's my sum. My expected, if this is my sum, and I'm using my 3:1 ratio, this is what I would expect. I can calculate that. This is my total, and so 1/3 of that should be wrinkled, and so 1/4 should be wrinkled, and 3/4 should be smooth. Figure the difference between them, observed minus expected. Then I square that number, and then sum it over here. And then I do observed minus expected squared divided by expected. So I take this number and I divide it by my expected number. And I have those two, and I add them up, and there is my chi-squared value. So remember, you're probably gonna have to do this in your exam, so practice. Now I have that number, I have a chi-squared value, and I have to use a critical values table, which I would have to give you, or it's also in the back of your textbook, and I have to know the number of degrees of freedom. So I had 2 categories, so I have 1 degree of freedom. Okay, my chi-squared value was 0.263, and my probability, my p-value is 0.05. Oops, what happened here? Okay, so my p-value is 0.05, and I have 1 degree of freedom. So the value, the critical value is 3.841, and this is quite a bit lower than that, so I'm good to go. This is the observed fit the expected. Okay, here's another one. So do this one as well, practice for yourself. and use the table. So pause the video, practice the question. Okay, so here's my yellow seeds and green, my observed. I take the sum of them here. I calculate the expected using my Mendelian ratio of 3 to 1. I take the difference, I square them, I divide by expected, and my chi-square value is 12.32. So look at my value, my table here again. I have 12.32 as a chi-squared value. We use 1 degree of freedom because I had 2 categories. Minus 1 is 1. Is it a good fit? Well, 0.05, 1 degree of freedom, my value here is 3.841. That's my critical value. This is much higher, so it's not a good fit. Okay, you can also use this for independence. There's only 2 chi-squared tests. One for the chi-squared test for goodness of fit and the chi-squared test for independence. So make sure you write both down in your notes before the exam. So for independence, I want to see if the 2 are related or not. So I have a contingency table in this case, a 2-way table showing frequencies. So there's 2 steps here. Okay, so this can only happen when I have at least 5 frequencies. All the frequencies, at least 5 or more, they're showing up. When the samples are taken at random from the population. And so that's why I had to keep this in mind before I'm doing my experimental design. I have to make sure I'm planning on doing chi-square tests that I have at least that amount of data happening. And then I'm going to calculate each expected frequency from the values on the contingency table, and I'm going to use this equation. So it's an extra equation. Because you don't have something like Mendel's laws to figure out the expected, you have to use an equation to help you do that. So the sum of the row times the sum of the column over sum total gives you your expected values. Okay, so here's a practice one. They counted the seeds on holly trees in a sunny position. And talked about look at how the different numbers of seeds inside a larger berry versus a smaller berry. So write your hypotheses. Null hypothesis, the 2 variables are independent. The number of seeds is not significantly different on the berries in the 2 trees. For the shade tree versus sunny tree. And the experimental hypothesis, the 2 variables are associated. There are more seeds in the berries on one tree than the other. So how do we do our test? Well, we make a contingency table. So here I have my observed values to be given to you. This is only observed values. So the trees in the shade, the number of seeds number of berries that had 2 seeds was 12. Number of seeds that had 3 berries was 40. Number of seeds that had, berries that had 4 seeds was 8. Here's my total. The tree in the sun, I had no berries with only 2 seeds. I had 17 berries with 3 seeds, and I had 43 berries with 4 seeds. There's my total, and I just do sum on the bottom. So to figure out my expected, I have to use this formula. So the tree in the shade is going to be 6. So I have the sum of the row, which is 60, times the sum of the column, which is 12, divided by the sum of the total, which is 120. And that for this one gives me 6. Same thing here, 60 divided by 60 times 57 divided by 120 gives me 20.5. I'm just using this formula. Okay, 51 times 60 divided by 120 gives me 25.5. And there's my total for that row. The same thing for the tree in the sun, and the total is the bottom again. So now I have my observed values and my expected values, and I can do chi-squared. So I've just made another table here showing how this is going to work, just to make it a little bit clearer. I have my observed and expected for both shade and sun trees. Okay, so here's my chi-squared. I'm going to do my observed. I have it from the table. So 2 seeds for berry, I had 12 of them. 3 seeds for berry, I had 40 of them. 4 seeds, I had 8. My expected, I had 6, 28.5, 25.5. My difference is here. I squared those differences and I divide by expected. So that gave me a sum chi-squared value of 12.01. Now my degrees of freedom is going to be figured a little bit differently when I'm doing this kind of chi-square test. So it's m minus 1 times n minus 1. So m and n are the number of rows and the number of columns. So I have 1, 2, so in my original table here, the contingency table, I have 2 rows and 3 columns. right? If I go back, yeah, 2 rows and 3 columns. Just zoom on through here. So if I have 2 rows, m times 2 minus 1 is 1, and 3 minus 1 is 2, so 1 times 2, I've got 2 degrees of freedom. So our chi-square value is 45.3. We have 2 degrees of freedom. So we can accept or reject. Well, if I have 2 degrees of freedom and 0.5, I have 5.991 is my critical value. So we reject. 45.3 is way above 5.991. So the variables are related. There is a connection between shade and sun and the number of berries. Here's another example of a question on the exam about using chi-squared, and this one they asked you to just describe how you would do it. On your exam, you will have to do chi-squared calculation, so make sure you practice. Okay, that was my review. Hopefully this is helpful for you guys to study for your exam. If you have any questions, you want a little mini Zoom convo about some of this, do some practice, please let me know. Good luck, guys.